shouldn't take too long, and it's going to be that way for the semester. Normally, the way it works for 1407 and the manual, the department lab manual I follow, um, it's a lot of what I call anatomy. Usually, most of the anatomy involves using the microscope, and doing drawings and sketches of specimens. So it makes it a lot more difficult to conduct these labs online. So some of these labs for some of you may seem super easy as far as what you have to do, but don't take for granted because you still have to make sure that when you complete the lab, that the lab will upload without any problems, okay? So this lab is going to be a combination of labeling some diagrams, answering some questions, okay? You can do it directly on the document. Actually, not to copy and paste the figures on a separate document and label them, but make sure you do not wait till the last second to do this lab. Because if you email me a week from today telling me I'm having problems with getting this document, the labels and all that to upload, I'm not going to cut you any slack, period. Okay, so make sure you have 2 attempts. Get on this early. By now you should know part of the challenge isn't just completing the work, it's getting the document to upload without any problems. Okay. Now we already did in the spring. We spent the last half of the semester 07 online. So and the students had a lot more labeling things to do, and the vast majority were able to get it. So I know it's doable. What we're doing. Okay. Let me go ahead and jump into this. So I don't take any more time. All right, today's lab, what I have here, should say lab 2. I'm sorry, I'll edit this. Should say lab 2. This is normally lab 1, and we do safety training overlapping. I'll fix this, but this is lab 2, and it focuses on evidence of evolution. Let me see if I can get this to go here. Okay, and it starts off with some background information about evidence for organic evolution. Must show that organisms have changed through time and that closely related organisms have some features in common. We talked about biology before already in lecture. So in this lab, you will look at 3 kinds of evidence that support the theory of organic evolution. And this basically falls, the first 2 basically fall under what we would call a combination of homology and fossil records. If you recall at the end of chapter 1, it talked about 4 lines of evidence, direct observations, homology, fossil records, and biogeography. So you can see that basically this is where all this falls under. So we have the fossils, which is the first part, very easy, very quick. Comparative anatomy may or may not be easy. Some of the figures I found that I'm using, they're hard to see, so I'll point out where do the best you can in labeling them, because you're going to be labeling bones of what we call the upper limb and bipedal organisms such as ourselves, which are upright, or the forelimb or the pectoral region for other organisms. And then the last part will be biochemical comparison of amino acid sequences and proteins. So basically, you're looking at 2 of the 4 lines of evidence that I gave you in lecture. Okay, you're looking at fossil records and then homology. And the biochemical comparison of amino acid sequence and proteins, they're going to talk about a protein, a cytochrome protein, that often you hear about in cellular respiration. Take a biochem course, and how they sequence this protein. Okay, and they use the homology of that amino acid sequence of the protein polypeptide and comparisons from one organism to the next to see how much homology there is or how different they are, and then they equate that to give them an idea of how long it's been since they've evolved separate from each other. So fossil parts is pretty. You have an Act 1, which is comparison of a fossil form to an extant form. Extant means living, and I'm introducing this word to you now. You'll see it again later in the chapter. But basically, I've given you 2 pictures, so you'll just do it on the picture. And what I've given you are fossils of what we call an ammonite versus a nautilus shell, cephalopods. Ammonite's if you want to call it, the ancestral form of a cephalopod, and the nautilus, I'm showing you the shell, the nautilus shell. The nautilus is a cephalopod that's a present-day living organism. And it's simple. You don't have to overdo it. Just point out, what these structures have in common based on what you can see. Are there any differences that you can also notice? Okay, don't try to overdo it or be elaborate. Just try to keep it simple, but give me a thorough answer. And just be one characteristic for each of these that you give me. Then it talks about examining common fossils, or what they call trace fossils. Okay, fossils come in many shapes and forms. While some fossils may be intact and include the entire structure of the organism, other fossils may be found in pieces. Now, this is misleading. Okay, entire structure of the organism. When fossils are discovered, it says in your notes, I believe I still left it there in your lab protocol itself, okay, the fossils are not actually the direct remains of a former living organism or formerly live organism, okay? It's basically a cast or a mold, okay, of that organism. But the tissue itself has already degenerated or decayed and is gone, but there's like a cast left or a mold left. Now, some fossils, however, are what they call trace fossils, okay? In other words, what we call like dinosaur tracks or worm tracings in the ground. So just answer those questions, give a few characteristics about what they tell you. Again, I'm not expecting some elaborate answer. It is meant sometimes to be that simple. Then the second part for the line of evidence is comparative anatomy, okay? You've been given some information that talks about how you're going to focus on the, sorry, let me go back. You're going to focus on what we call the upper limb, the forelimb, the pectoral fin, okay, or the wings of organisms. And what it's going to ask you to do in that second part for comparative anatomy is it's going to say, hey, look, you're going to focus on the bones of what we call in lay terms the arm. In anatomy, okay, if it's a two-legged creature, we say the upper limb. If it's a four-legged animal, we say the forelimb. If it's a, what we call a fish or a dolphin or a whale, we call it the pectoral fin. Okay, in any case, these bones you will find in these animals. So what I've given you, okay, are either pics or actual pictures of the skeleton, or at least that part, that appendage of the skeletal bones of the organism, and I want you to label them. But it tells you in the protocol when you label, okay, the bones that have been given of different animals. All right, it says you don't have to rewrite the whole name of the bone, such as humerus. You can just use H, radius, R, ulna, U. Now, what I've given you here is a skeleton, and I know it's not the greatest. I thought I had a better one because I taught intro to A&P. But this is what you want to look at, okay? You can start with the human arm, upper limb, I mean. The human upper limb is what they call it in A&P. Just look at these bones here to give you an idea of the relative position, especially the bones of the forearm. In anatomy, we call this the forearm. Up above it, we call it the arm. And this whole thing is called the upper limb for us, for human beings or two-legged mammals. Okay, the bones of the forearm are usually the most difficult ones to ID because you have two bones there, the radius and the ulna. And what I used to tell my students is the way you find the radius is start on the right limb if you can, or simply locate what looks like the thumb. Okay, and in what we call a prone position here, okay, the bone that's running on the outside towards the thumb, that's the one that's going to be considered the radius. The one that's going to be located inside or running along in the direction of the small finger, that'll be the ulna. Now, some of these will be hard to tell, so just do the best you can on those, the figures that I gave. And like I said, you can just, I uploaded it as a Word doc and as a PDF, so see which one you can work with easier. And these are some of the organisms here, so you can also use the PowerPoint to kind of help you narrow things down. I'll let you review all that. The last line of evidence, okay, to support evolution, okay, is molecular evidence, and it's what they call a protein clock. It says molecular data is often used to determine relationships between different organisms, particularly comparisons of DNA and protein. What you're going to be looking at is protein comparison, specifically the amino acid sequence of the primary of a protein. Okay, now everybody's supposed to know what the primary structure of a protein is. If you don't, that's up to you to look that up, okay? What I mean by primary structure of a protein. Now, the various proteins found in organisms are made from patterns that are stored in the DNA. As I tell my students, the genetic information you inherit from your mom and dad, okay, a large majority of that reads like a recipe book, okay, that tells the cell how to make proteins, and proteins do the work in your cells. Therefore, we would expect closely related organisms to have similar protein structure and distantly related organisms to have more differences in their protein structure. This is what's going to be asking you to focus on. The greater the difference, okay, in the amino acid sequence, when you compare proteins, what's supposed to be the same protein, but for two different species, the greater the difference, then the longer they've evolved apart from each other. Okay, but when you look at the amino acid sequence of the particular protein they gave you, the higher the degree, okay, or the more significant the homology between the two, then the way a scientist views it is these two species, even though they're separate, they more recently underwent speciation. So they're still closely related. Now, the protein clock theory, it states that the number of amino acid changes between proteins in two organisms is proportional to the amount of time that they have been evolving separate as common ancestors. So this is a key phrase here, this protein clock theory you're going to need to know going into answering the questions, okay? And then what it's going to give you is it's going to give you, I just put that there, but what it's going to give you is this table here, okay? Now, you may not be able to see it here clearly, but it's in your lab. All right, should be, I believe, second to the last page. Notice the animals that are, the organisms, sorry, not animals, because there's also unicellular organisms. The organisms that are listed here are the same organisms that are listed here. So there are questions you're going to answer. Answer every question on that last page. Give me a thorough answer. Okay, don't give me an answer and you assume I know what you mean. Explain your answers. Explain your reasoning like it asks you to do. Okay, and what it's going to do is basically it'll give you two animals. So for one of the animals, you'll look them up here. For the other animal, you'll look at them here. And then you'll just follow the column and the row to where they meet. The number they give you, that tells you the number of amino acids that are different between that identical cytochrome protein, or that cytochrome protein that's found in both organisms. And then you just go back to the protein clock theory and what it tells you. The greater the number of differences, what does that mean? The fewer the number of differences in amino acids that make up the protein, what does that mean? And then make sure you answer all those questions on the last page. Simply put, you can label them on the handout that I gave you. You can use text boxes on there. Some of my students, if they don't mind using the printer ink, they'll print it off and they'll handwrite it in there. That's fine as long as I can read your handwriting. And then they'll scan it and upload it back up. That's fine too. Okay, but do not wait till the last second and then send me emails or contact me saying, hey, I'm having trouble getting it to take or upload. Get on this early, okay? Because you're going to have a lot of labs like this where you're labeling diagrams and pics that I give you. Are there any questions? I know it's short. It's not very long. It's going to be that way because, like I said, normally I don't say much. I don't have to say much. Normally students do the work. They're just sketching drawings and things of that nature and labeling. No, no cover page. The PowerPoint, is the PowerPoint available, everybody? Nobody looked. It's not available. I didn't post it. Hold on, I might lose y'all. Oh, wow, I did screw up. Okay. All right, Cheryl May, thank you for asking me. Boy, I tell you, I'm getting my wires crossed up with all my classes. Thank you for that question. Yes, the PowerPoint will be available. Okay. It will be available. When I get out of here, I'm going to put it up. I apologize. Thank you, Cheryl May, for asking me. I normally put it up. Sorry. I owe y'all one there. I'll remember that as I continue making the exam, okay? Be a little nicer. Any other questions? Oh, before you leave, one more thing before I stop the recording too. If you didn't read Blackboard, I mentioned on Blackboard that under syllabus, I placed a link to Office 365. Okay, if you're like me, tech challenged, go under syllabus, find the link. It's just titled Office 365. Click on it. It'll take you to an ACC web page, okay, that'll give you some information. Microsoft, I believe it is basically pushing everybody to use the more recent versions. The reason for it is because of all the problems with hacking and what have not and viruses and things. So what ACC has done is they're making Office 365 available for free to employees and students. So please take a look at that if you're not familiar with that. I put the link under syllabus, okay? All right, I'm going to end it right there.